It is sort of like being a kid in I mean, it's changed everything for us.
Okay, let's bring it out of the laboratory now. Let's go to Dr. Borma here and find out, as a plant breeder, all that he's finding and others are finding now in this sequencing and in the lab. Let's bring it out to plant breeding. How's it going to help?
And Mike, it's my view that the availability of the soybean genome sequence is basically allow us as seed breeders, I like to say, do more, do it at a reduced cost, and do it faster. As an example, in the past 35 years, we've gone from it requiring 11 years to develop a new variety. The use of winter nurseries took us down to 8. The initial markers that we have taken us down to 5 or 6, and now major seed companies are talking about less than 5 years, down to almost 4 years in the varietal development process.
So in comparison, even though it still takes a little time, because you've got all this to go through, it's a quantum leap from what we were before.
Yes, sir. For example, there are a couple, for example, a Chekhov-sponsored project to improve the fatty acid profile requires bringing together in an elite variety 6 different genes. And in the past, it had almost been an impossible task to achieve that on a wide basis. And this new sequence is going to greatly hasten that. Another thing that we're allowed to do now, or are capable of doing now, is with these various herbicide tolerance characteristics or traits that are available, many breeders are wanting to pyramid them, have more than one in a given variety, and it's going to greatly enhance our ability to do that. Other thing, when you think about the speed of the process, it used to take in a greenhouse 35 days to do a cyst or root knot nematode resistance screen. And today we can do that in 4 days or less, and actually at 1/10 of the cost. And kind of my favorite look at the thing, and it's a little longer range, but we aren't able to do it now, is breeders like to do a lot of forward crossing 2 elite varieties. When we do that, out of these 65, 66,000 genes we're talking about in soybean, probably in any one cross that we make, we're probably seeing 10 to 12,000 of those genes segregate. And what breeders like to be able to do is select for these hot spots in the genome, and this technology will definitely empower us to do that.
Whoa, a hot spot in a soybean? Now, what are you talking about here, a hot spot?
I was afraid you'd ask me. Really, it's basically what we're talking about is there are certain parts of the genome that have just been present in all of the incredibly useful cultivars. And so there are a number of these spots in the genome, and breeders, when they make that cross, can make certain that those different regions are fixed before they do any other selection. So it's a very, very empowering tool.
Now, all of this is what we're finding now is already in the soybean. I mean, it's not like you're adding something, have to find something. It's in there.
Yeah, it's true. What's the difference, though, is, you know, we've been developing varieties in the United States since the late 40s, early 50s. But what we're able to do now with these technologies is actually pack more technology, if you will, more desirable genes into one variety and make it much more beneficial for the American soybean farmer.
Whether it be drought resistant or whatever trait you're looking for. And when we come back in just a moment to both of you, I want to get to where you see this going, where this could take us. But first I want to bring in Rick Stern. Rick, soybean farmers made this decision. The soybean checkoff was going to fund this type of research that has brought us to this point of sequencing the soybean genome. Why did you feel it was so important?
Well, the checkoff continues to invest farmer dollars effectively and efficiently, and this is one of the many areas that we saw many years ago. The past checkoff leaders looked into the future and said soybean genomics was one of the many areas where we're going to bring farmers to be effective and efficient in a global marketplace.
So you could see that this was a direction that was going to bring benefit to soybean growers and the potential that was there to unlock that soybean.
Yeah, we started in 1995. The checkoff funded a few basic projects in soybean genomics. And then in 2003, we hosted a genomic strategic planning meeting and brought all the main players together. And out of that meeting, the scientists said that this was an area, basically building the genomic map was an area of improvement for soybeans in the future. And then in 2004, the checkoff funded 3 basic projects beginning to build the physical map, which basically put the checkoff in a position to have projects that were looked at by the Joint Genome Institute and the National Science Foundation, which leveraged this to where it's at today.
Let's look at where we're going from here. Dr. Cregan, where are we going with this?
Well, as I said, we've been developing these markers for many years, and now probably in the next 3 months, we'll develop another 50,000 markers. And as part of a checkoff project that we began working on actually last year in anticipation of the genome sequence, we have a new scientist in our group at Beltsville, Dr. David Heighton, who will work in collaboration with Randy Nelson, who's the curator of the soybean germplasm collection. Soybean germplasm collection, the USDA collection at Urbana, Illinois, has approximately 18,000 soybeans that have been collected since beginning in the 1910s, something in that range, have been preserved, grown out, and maintained, and all kinds of data taken on them. What Heighton will be doing in our laboratory is isolating DNA from all of those and characterizing them with these 50,000 DNA markers. So we'll be able to then look at the different forms of genes that are present in order to discover different resistances to phytophthora, for instance, or rust, or different protein oil levels that we're interested in. And there's just no data set in any species like this. The whole germplasm collection, 19,000 soybeans. And we will also do other elite cultivars that have been released over the years as part of this project. So it's just an amazing project, and I think ultimately, as you were talking about portions of the genome that have to be there, we'll be able to determine in great detail what are those portions that we want to put together. Pyramid is a term you use to make ever-increasing soybean productivity. So we're obviously, I'm pretty enthused about that.